and it is a focus of the area is plant disease diagnostic and plant microbe interactions. Today he will tell you about the pathogenicity genes related, like sir is basically working on host pathogen interaction. So I think you can have the best glimpse here in the what we are doing in plant pathology about host pathogen interaction. He has before that he was in CSI system, then he moved to the ICR, and MSc from Kerala Agriculture University and PhD from Tamil Nadu Agriculture University. So one of the best pathological scientists in the area of plant interaction, host interaction. I think you can have lot of interest in that particular area. So my request is there, you ask the question as much as you can so you can have more understanding about that. Okay? So, sir, floor is yours.
Thank you. Sorry, I don't know what it is. This one pointer console, pointer, and then this is this one, no? This one or the one? Take it. Okay. Okay. Good afternoon, everybody. Thank you, Dr. Deepa, for the introduction. So originally my lecture was scheduled for 31st. I have forgotten to change the date. Later on, since I will be on a tour, official tour, I requested for a change and it has been rescheduled today. So, as told by Dr. Deepa, basically our lab is mainly within plant pathology, focusing on the molecular basis of host pathogen interactions. Okay. So, before I actually go into the topic, I'll like to give you a brief introduction about the basics. So, the pathogens normally in the absence of the host, they will be surviving on the debris or in the soil, and then it will be by dispersal mechanisms, various mechanisms, the pathogen propagules will reach the host. So from in the host, the pathogen has two types of interactions. That is, either it can be a endophytic, that is, it will be having a symbiotic relation, or it can be a parasitic relationship. So thereafter, again, during parasitic or whatever may be the interactions, they try to produce sporulation, reproductive structures are formed, which will be carried forward, again reach the debris or the soil. So the cycle keeps on repeating as shown here. So from survival to survival, dispersal and infection will be in between this. So during infection, as I already told, they can be of different types. Either they can be a parasite or they can be having a endophytic relationship, symbiotic relationships, something like beneficial one. So pertaining to this topic, I will be restricting myself to parasitic activity, that is the pathogenic activity. So I am not dealing about the other one. And then while talking about the pathogenicity genes, I will try to say what is its significance and how this can be exploited for fungal disease management. Okay. So now, like nutrition, like for we human beings, we have the nutrition and several other physiologies. The fungus also have many physiologies. One of them is nutrition. So nutrition in fungi is definitely of two types. Either it can be a biotrop or a hemibiotrop. Biotrop means it needs a living host. So it does not kill the host. It tries to have a living relationship. When the host dies, this also will die. So it does not want the host to die. Okay, it will be having a living relationship. So the molecular mechanisms during infection, as I told you in the disease cycle during infection, the mechanisms are different for a biotroph as well as for a hemibiotrop, a necrotroph. So necrotrophs are not very sophisticated like biotroph. They are not very intelligent, but they are very brutal. In the sense, they kill the host and live on the dead tissues. Okay, so this is the thing. So if you start see from the survival to survival, it will pass through the host. During the host interaction, it can have either beneficial or pathogenic, and in the pathogenic it can be either biotrophs or necrotrophs. So Thereafter, now this fungus during infection, now it has to, it will be subjected to various physiological factors, the temperature factor during the, when the fungal spore lands on the host surface, the temperature, oxygen, the oxidative stress, and then the host signal molecules. Everything is an environment to the pathogen spore. Okay, so it has to react according to the signal molecules. The signal can be a temperature, pH, or whatever may be. So once it perceives the signal through a signal transduction process reach the nucleus and then activate the appropriate genes required for that environment, either temperature or for pH or oxidative stress, or to the host signal molecule, which can be phenolics or so many other things also there. So this is how it happened. Similar way in the host also, in response to the pathogen, the host will be recognizing the pathogen molecules, and then there will be a mechanism inside the host, and then it will be revealing that phenotype as resistant or susceptibility depending on its genotype. So that I will explain it in the next one. Yeah, see here. So those who are having a pathological background may be knowing it, but for others, I don't know whether you will be knowing it or not. So when the pathogen lands, you perceive this as a host. When the pathogen lands on the host surface, the molecules are recognized by the pathogen. So whether the pathogen is, whether the host is susceptible or resistant, it's immaterial. The surface molecules are recognized. There will be a defense mechanism, which happens both in resistant and susceptible, which we call them as basal resistance or pattern triggered immunity. So thereafter, the pathogen will try to inject some molecules to bring down the resistance, otherwise it cannot colonize. So these molecules are the that reaction due to the injection of the molecules is called the effector triggered susceptibility, ETS. And then if the host is having any origins for these particular effector molecules, then what will happen? They will recognize these effector molecules, and then it leads to a higher resistance, which we call them as effector triggered immunity. So effector triggered immunity is greater than PTI, and they are mostly governed by major genes. Here it is mostly governed by minor genes, and this one is the effector triggered susceptibility. So this cycle keeps on repeating. So now what will happen? The pathogen has to overcome this, and again it will mutate and it try to produce a different molecule. You can see here red color, here it is producing a blue color molecule, bring down the resistance, and due course of time the host also develop a new R gene. So again this cycle keeps on repeating. Now in the context of our presentation, so the point here is these pathogen molecules which is causing ETS, and the genes coding for these molecules, we can term them as pathogenicity genes. Okay, the genes which are responsible for breaching the host barrier, okay, to gain infection and to manipulate. Now what is the difference between a pathogenicity gene and an effector molecule? Pathogenicity gene is a broader term. As I already told, in case of biotroph and necrotroph, it is same here. The molecules will be recognized, there will be a PTA mechanism, but in case of biotroph, they don't want to kill the host, so they don't produce enzymes or toxins, or they produce very, very less amount. So they try to inject some molecules which manipulate the host system to favor to the biotroph. Whereas in case of necrotroph, the molecules will be very brutal. They will try to break the cell wall, they try to kill the protoplasm, and then it will try to thrive on it. So this here, based on the nutrition, the mechanisms will be varying in the ETS mechanism. So this is the basics of this. I don't know where is this. I cannot see this. Okay. Now, so I hope you understand the context of our lecture. So in the life cycle, where plant will come, and within the plant where the pathogens will come, pathogenicity genes will come. That is in the ETS part. And what is the difference between pathogenicity genes and effector? Pathogenicity gene is a broad term. It covers everything, all the molecules, whereas effector is a proteinaceous molecule or some toxin molecule which manipulates the host system. It won't like destroy or degrade, it will just manipulate it for its favor, something like that. Okay, so you can say effector is a component of the pathogenicity genes. Okay, now how we will classify these pathogenicity genes? As I already listed here, the pathogen has to land on the host surface, and then it will get attached to the host surface, and then what it will do, the spore will be having some internal nutrients. With the internal nutrient, it start producing a germ tube, and then later on it will recognize the host surface, host signal molecule, and then it will start producing aprosorium. Aprosorium is a enlarged structure of the germ tube. Spore, germ tube, and aprosorium. And some fungus, they produce cushion shaped structure also. The aprosorium, cushion shaped structure, so they all vary depending upon the pathogen. I don't want to make it more complex, I am just making it very simple. So for these earlier events, several genes are involved. So we can group them as earlier event genes. And then the other one is to breach the barrier, to break the barrier and gain enter into it. That is the genes responsible for this. And the third one is the host will also try to react some defense mechanism. Like PTI I told you, that has to be overcome. So suppression mechanisms, the genes responsible for that. And then once the pathogen causes infection, it will spread throughout the plant, and finally it has to come out of the plant. How it will come out? As reproductive structures, which can be either first, normally they like to go for asexual reproduction, which is easy method for the fungi, and then under certain conditions they also go for sexual structures, both male and female, depending upon the classes of the fungi, and then sexual spores also will be produced, and again it will be carried, got, and then survive. So as I already told, so those certain mating type genes also act as a pathogenicity genes. Not all mating type, certain mating type genes, like in case of Basidiomycetes, certain mating type genes play a role. Then there are the pathogen genes of unknown function. Now this is what very important in the context of our lecture. See, because of this genome sequencing, you are getting innumerable number of genes, but many of the genes you are not able to annotate, or even if you annotate, you have to functionally validate what is the role of this gene. So if you are not able to functionally annotate the role of these genes, we can classify them as the unknown function genes. And finally the signaling genes. Signaling means what? The genes, how they perceive the signal, the pathogen signal, and then how it will react. So these are the various categories. I am not going into the details of each and everything. Our aim is, so where pathogenicity comes in the host pathogen interaction, and how it can be exploited for our management, fungal disease management. That is the purpose of this lecture. So as I already told, in the genome, due to extensive genomic sequencing of various organisms including fungi, a large number of data is generated. So data say that up to 40% in the model fungus Neurospora is not annotated. You know? Not annotated means they don't have functions. What is the function of that 40% gene is not known. But why extensively in fungi genome sequencing has been carried out? It has its own advantages, like compared to other eukaryotic organisms like plants or animals, it can be very easy to grow in the lab, and then the size is also relatively small. The smallest genome size in plant is Arabidopsis, which is roughly about 125 MB, whereas the average size is 30, it may go up to 85 also. Some Oomycetes are, that is fungi-like organisms, they have little bit higher one, but average I am saying 30 MB, and also they are haploid. So haploid has its own advantage and disadvantage. So because of this reason they are very well sequenced compared to other organisms. And then one of the in silico approach is how to identify the pathogenicity genes by in silico method is this PHI base. There is one website where we can go for identifying the pathogenicity genes of different plant pathogen systems. Now, okay, we have got this genome sequence, we have got many genes, but we could not predict the functions of all the genes. That what could be the reason? Because the genes may be redundant. Redundant means when they are associated with other genes, they may not be having any function. Okay, when they are alone only, they will be having function, or the conditions in which we are testing in the laboratory condition may not be suitable for the gene to express, and so we are not able to identify it. And then other thing is, most of the gene function annotation in the pathogenic fungi we are doing based on comparison with the model fungus, that is the Saccharomyces cerevisiae, which is well characterized. In the previous lecture you might have heard about yeast genomics also. So maybe in Saccharomyces it is not annotated because of that reason. Here also it is not annotated. Now the vast array of genes, they become useless if they are not functionally validated. You need to functionally validate it. Functional validation means you assign a function to the gene. So how it can be done? There are some functional genomic approaches are there, which we broadly classify them into random mutagenetic approach or targeted mutagenic approach. Random mutagenesis means the host organism, you mutate it randomly, you don't know what is that gene, and then once you develop the mutant you have to characterize it to identify the gene. Okay. Targeted means you know the gene, and for particular gene you will be trying to develop the mutant. Okay. So the various factors which are favoring. But what is the advantage of functional genomics in case of fungi? It is having a better transformation system. It is easy to do transformation because every every cell or tissue is amenable for this one, and then both this random and targeted mutagenesis is very much feasible. But in case of plants, random mutagenesis is very tedious. Like if you are going for tDNA mutagenesis, you will get the genome size is very big, so large number of mutants will be there. Every mutant you cannot screen to find the function. Whereas the genome size is less, relatively less, so we can get lesser number of mutants, so easy to characterize them. So this is one advantage. Now, generally in functional genomics, what is the approach? The forward genetics and the reverse genetics. To make it very simple, from the phenotype to the genotype is the forward, from the genotype to the, if you know the gene and what is the function of the gene, that is genotype to phenotype, that is the reverse genetics approach. Okay. So, but either cases, either forward or reward, you need to develop a mutagenized population, which can be either natural variation or you can induce them. So, you get the phenotype. Okay. So, then that phenotype to genotype, identifying the gene by mapping is the forward genetics. It has its own advantages. It is objective information regarding a mutation. Objective means what? Unbiased. It is, there is unbiased, but you can clearly say, pinpoint out this is the reason. And then you don't need any knowledge about the gene also, and it has its own disadvantages. As I already told, in random mutagenesis you have to screen a large number of mutants, and that will be very laborious. So the next approach is the reverse genetic approach, which is the very common one, very commonly followed even in plants also, as well as in fungi also. Because we are agricultural students, so I have to quote everywhere. Whenever I talk about fungi, I need to compare it with a plant, because plant genetics is well studied, well known, mostly we know it. So fungal genetics is relatively less studied and we are not aware of much. So whenever I have to explain it, I need to take the example of plant genetics or plant whatever related to plants, and then I have to see what is the difference or what is the similarity. So in case of reverse genetics, as I already told, from the, you know the gene, you try to mutate the gene either by silencing or editing, various approaches are there, which I will be telling very briefly, and then you try to see the phenotype by developing that mutant, whether that mutant is able to sporulate, conidiation, whether it is causing pathogen, disease, infection or not. So you are studying the phenotype. So from the genotype to the phenotype. So it has advantages. It's less time consuming when compared to the forward genetics, and you don't need the knowledge about the genes in this case. Gene function. You should know the knowledge about the gene. Because without knowing the gene, you cannot design the construct or whatever may be. But gene function is not required. The disadvantage is single allele mutations. Sometimes they will be very misleading. You may get the mutant, but the function may not be there. So why reverse genetics is very common in fungi? What are its? So as I already told here, there are several advantages. You can read it here. And then this is very much feasible in case of necrotroph and hemibiotroph. Okay. Now what are the requirements to develop to investigate the gene function? The various requirements should be, you should be able to have a better transformation system, because in fungi also in plants, so many plant species are recalcitrant. You cannot develop a transformation system, So, but in case of fungi, mostly they are amenable for transformation, and you need a better marker and a mutagenesis technique. Now, what are the various methods available? You can see here the various methods as I listed here, but most of these methods, they depend on protoplast. Okay, so this is one disadvantage. But otherwise, it's very much easy. But you need a protoplast. Protoplast, you have to handle it very carefully. The very common method is in our lab or everywhere they are following is the calcium chloride polyethylene glycol. So as I already told, this is the disadvantage, protoplast. Then what are the marker genes? Why we need a marker? So you need to distinguish the transformant from the non-transformant. You know? So you need a marker system. In bacteria, you know, ampicillin is kanamycin, all the things very commonly used, some like that. In eukaryotes, plants and all, the most common marker is the hygromycin. So I have highlighted here. Besides that, you can also use like this based on this visual distinction, GFP proteins, Gus markers, and then there are some nutritional genes also, as I shown here, like this. In our lab, we use this herpes simplex virus thymidine kinase. So what does this mean? It's a negative selection marker. Normally, the positive marker means the transformants will be selected in the presence of the chemical or whatever may be, whereas in this case, in the presence of that one, the transformants will be dying, they won't survive, because the thymidine kinase will utilize this analogs, and then it will cause death of the transformant cells. So this is a negative selection marker which is also very commonly used. Now, so in some remote cases we have a combination of either of this like this visual distinction, GFP plus hygromycin. So there will be a combinations also, that depending upon our work and the nature, our interest, we can select the marker genes. And the type of mutagenesis is the random mutagenesis technique and the targeted mutagenesis, which I already told. So what is a random mutagenesis? The DNA which you are, the construct or DNA, whatever may be, which you are inserting into the host genome, will not be having any homology with the host. Okay, this is the major point. As I mentioned it here, there are various methods are there, like REMI. REMI is a restriction enzyme mediated integration, DNA tagging, transposon tagging, and TILling is targeted induced localizations in genomes. So these all the methods are followed in case of plants except the REMI. REMI is done exclusively for fungi, whereas other methods is very common, random mutagenesis techniques in DNA tagging, transposon tagging, and this TILling. And TILling is very, very common among breeders, you know. Now what is REMI? So in case of REMI, you have the construct. So here you have the construct, the whatever may be that one, and then along with that you will be also using the restriction enzyme. So the restriction enzyme plus construct, you will be transforming into the host genome. What will happen? The restriction enzyme will make a cut in the host genome, and then this construct will be actually in case of fungi, unlike bacteria, we have to linearize them, make it straight, because the efficiency will be more. So the linearized construct plus enzyme you are transforming into the fungal genome. So this enzyme will cut the fungal genome, and then this linearized one will go and sit here. By that way you can develop a mutant. So it has some disadvantages. As I already told, protoplast is very cumbersome, and then there will be significant different so this integration also may not be very precise. It will be for different wherever the enzymes have that matching recognition sites are there, they will keep on making the flick, so that there will be non-homologous integration also will be there. Then tDNA tagging. tDNA tagging is very common in plants also, it's very common in fungi also. The same thing like that, you know, what is tDNA, agrobacterium, all those things. So now this when you are integrating into the host genome, the fungus genome, what will happen? It will randomly go and integrate. The tDNA will go and insert itself in different regions of the chromosome, and then what will happen? They will mutate it. How to identify the mutant? By tail PCR. Okay, and then ultimately it will leads to what? Knockout, knockdown, or knock-on. Okay. Then the other method is, this is the one picture which I have shown, the fungal cells with Agrobacterium tumefaciens, it is having the tDNA randomly integrate. Then this TILling. TILling is nothing but you have to develop a population by using a mutagenic agent. The mutated population, then you have to see where the mutation happened by various techniques. One is the very common method is the cleavage by restriction endonuclease, that DHPLC, denaturing HPLC, and high throughput sequencing. And then you can characterize the mutant. The only thing is, after developing the population, you have to pool all the DNA. That's a pooling will be there. Library you have to make it, and then you have to find where is the mutants are there, and then various methods, as I told, you can use them. The next one is the targeted mutagenesis. I am not giving very minute detail because time is also constrained. It's very elaborate, and you may not be able to follow. So the salient things I am trying to highlight it. In the targeted disruption, what you will be doing is the particular targeted gene will be disrupted or deleted. Okay. So how it can be done? So you need the construct, whatever construct you are making, linearest one. So you know the structural gene, there is a 5' UTR and this 3' UTR, 5' untranslated region and 3' untranslated region. So that region, for that I can show you the picture here. You can see this. This is the target gene. So this is the 5' UTR and the 3' UTR. What you have to do, you have to design a primer to amplify this prime with two primers and another set of primer. So you get the homologous region. Then you have to separately from the another construct where the restriction, this your antibiotic resistant gene is there, you can amplify them. So all three you have to integrate. This is the one what is this? That 5' UTR, 3' UTR, this is the antibiotic resistant gene. So now you will get a construct, linearized construct. This you can transform into the fungus, and then this region which is homologous, it will try to find the homologous region in the gene, and then they will try to mutate it. So your gene, the target gene will be either disrupted or displaced depending upon the cases. So this is the thing, the length of the homologous sequence. The length of the homologous sequence means how much your that flanking region is homology with the target genome. So that's the point. Then the level of homology. So level of homology means how much similarity between them they are having it. And then what is the transformation method and the genomic position all plays a major role. Okay. So this is what called the homologous recombination. But in case of plants, this is not very common. In homologous recombination, either you can go for a simple PCR, fusion PCR method, or you can use the plasmids, the simple plasmids. But in case of plasmid, what you have to do, you have to use the restriction enzyme. When you design the construct, when you amplify the gene, you have to use the primer with restriction site, and then you have to digest them, then you have to clone it in the vector, and then you have to do it. The principles are same in both the cases. Both are component of the homologous recombination. Both fusion PCR and ligation mediated PCR are two methods within homologous recombination. Then comes the RNAi. Initially it was evolved in animals, and then which we call them as RNAi, and then PTGS or co-suppression in case of plants. Quelling in fungi it was first termed as quelling, but nowadays very commonly they are using as silencing. Now this is a very precise knockdown. Knockdown means the gene will be expressing into a protein, but at a lowest level. Unlike editing, where it will be totally displaced, here it will be knocked down instead of knockout. So the principle is, I hope you all know by this time. So whenever when you insert a double-stranded RNA, either naturally it happens by virus infection, or artificially you can introduce by making a construct. So this double-stranded RNA will be restriction digested by the dicer enzyme into this much nucleotides, and then they will form a complex with the a special type of protein, arginine protein, to form RAC complex, and then they target the gene. So by that way, it has some advantages over the antisense traditional methods. You know that flavor saver tomato, again this is a polygalactonase inhibiting protein, which was first transgenic plant commercially, in the way back in 90s. So that was by antisense technology, but RNA has its own advantages. You can see here what are the advantages. Now, so these are all theoretical things, so I am just skipping it. I will go to the next method, is the CRISPR. CRISPR method, again, what you need a guide RNA. Guide RNA should be having homology with your target gene, and then the guide RNA with the enzyme, the nuclease enzyme, which is able to cut the target DNA. So both, that which is commonly used one is the Cas9. So Cas9 is the nuclease and the guide RNA. The guide RNA will be having homology to the target. So both you will be transforming it, and based on the complementarity it will go and edit it, and then you can develop a mutant. So as shown here, this is the complex Cas9 nucleus with guide RNA, and this is the host genome. Now the point here is, we often think that CRISPR can be used only for replacing, only for mutating, but it can be also used to replace it with a desired gene also. So these are the briefings about the various techniques, random and targeted mutagenesis of pathogenicity genes. So why we have to do? Because there is a large category of genes which are unknown, whose function could not be annotated or could not be validated. So those needs to be, we have to validate them by developing a suitable technique to mutate them. So whichever, depending upon the pathogen or fungus, we can use the technique. Okay, because certain fungus are not amenable for tilling, certain fungus are not amenable for editing, so you have to select a suitable method. Now, the purpose of this work is how we can utilize in our day-to-day activities for translational research. So, what in our lab, again I skip it, all those things, I'll just come to the main point. The main point is we try to find the role of these pathogenicity genes in physiological races. You know what is a physiological race? Physiological race means they are cultivar specific, okay, variety specific, you know. So, do the pathogenicity genes have a role in physiological races of Fusarium species infecting tomato? That we established by routine studies, okay. And see, these are the three effectors. See, this Fusarium oxysporum, which is causing vascular wilt in tomato, secrete three types of effectors. 6 means secreted in xylem. 6,4, 6,3 and 6,1, and the matching R gene is I1, I2 and I3. So we determined the role of the pathogenicity genes in race profiling. What we could found was that the pathogenicity genes are not good in, not all pathogenicity genes are good in profiling these fungal races from fungi into different physiological races. That is our final observation. Later on, we tried to develop unclassified, not annotated genes. We tried to find the function. So for that, what we did, we made a construct, fungal silencing construct, and then we transformed the Fusarium protoplast with that construct. So it will silenced the particular target gene, and we developed the mutant, and that mutant. So this is the method of making the construct, and then we developed the mutant, and the mutant showed less mycelial growth, then less conidiation and less pathogenicity. So once we validated the role of the genes, two genes, that is FOW1 and FOW2, which are mitochondrial carrier protein, we tried to go for host-induced gene silencing approach. So what we did in host-inducing gene silencing approach, same target gene, same pathogenicity gene of the Fusarium, we tried to develop a silencing construct, and then for the plant instead of the fungus. In the previous cases we did for the fungus, here we did for the plant, and then we transformed into the plant. So when the fungus, which is a hemibiotrop, infecting the plant, hemibiotrop means they have an initial biotrophic phase followed by the necrotic phase. During the biotrophic phase, the siRNA molecules were uptaken, and then they were the target gene was silenced, which we confirmed by all these methods, and then they showed reduced pathogenicity also. So the colonization also has been studied. So to summarize, we used RNA silencing method in the fungus and host-induced gene silencing approach to validate certain uncharacterized genes in Fusarium oxysporum. And then later on, we also tried to find the role of the genes in response to the plant signals. We could found that the genes are playing a major role in the plant phenolics, the earliest known signal molecule. One more work which I like to highlight is, very briefly, the southern corn blight in case of maize. There are two types of pathogen that races are there. One is the race T. Race T is able to produce T toxin. The T toxin is highly virulent on maize lines having TMS, that male sterility, cytoplasmic male sterility. The other race is the race O, which does not produce toxin, and it is pathogenic both on T and N. So what we could found in this study is, so you see here, as I already told, the model fungus or the yeast, that Saccharomyces, in that they have identified one yeast activation protein 1, YAP1, which was responding to the oxidative stress. So similar to that one, we found one activation transcription regulator protein, which we call them as CHAP, that is Cochliobolus heterostrophus activation protein 1. We named it as CHAP1. And then this CHAP1, what will happen whenever there is any oxidative stress, this protein from the cytoplasm moves into the nucleus and trigger the antioxidant genes. Okay, so we tried to see whether this CHAP1, how it will react to the phenolics, because apart from oxidative stress, phenolics are the earliest known signal, small molecule phenolics, not the major one. So what happened is, when there is a phenolics, what will happen? This transcription factor, this protein moves from the cytoplasm to the nucleus, like in case of oxidative stress, but they do not trigger the antioxidant gene. but they trigger some phenol metabolism gene. So at the end of the day, after this work, what we could found was there are two types of. This is the summary of that work. So the pathogen recognized the force signals by two signal transduction pathway, one for oxidative stress and the other for the phenolics in a different way. That's it. So what I have explained is the basics of infection and the pathogenicity genes, how this can be extrapolated for translation research for our day-to-day work, particularly in agriculture for fungal disease management, so that it can be useful to the farmers for enhanced crop yield. Thank you. Thank you to the organizers, to Dr. Kumar and Dr. Deepa and all who are involved for giving me an opportunity to share my experience and our lab work also to this. If you have any doubt, you can please ask me. I know I could not go very detailed due to time constraint, but if you have any doubt, all are published. It's available in the public domain also. You can go through it if you have any doubt. And I'm also available in the division also. You can always come to me and ask.
Yeah. Any questions from anybody? So some pathology students must be having some doubts. No? Yeah, stand up here, please.
Thanks for the information, sir. Sir, what would be the average turnover time for creating mutant fungal mutant, targeted mutant?
Average?
How much time it takes?
Turned out time.
No, I'm not. You are asking how much time,
How much time it
to
takes?
takes to develop a mutant, or it will be retaining it?
No, sir, actually to develop mutant.
So how much time it takes if you do this research?
That is
That's
a
very
question. So it depends upon your expertise and the facilities available. How I
simple.
can say?
If you know how to drive the car, you can reach the destination very fast.
How
You
it
do not
is?
know, it takes time.
For a beginner, it will take a lot of time, 2 to 3 years or 4 years also. But once it is done, then it will be.
Anyway, it's not a very path-breaking question. Anyway, still, you come to the main point. You are a PhD student, want to complete everything on time. Good, I appreciate that. Anybody else?
Mostly this kind of work we don't give to student, we do in project work.
Stand up.
How is the dsRNA given to the fungal pathogens?
So there are two ways are there. Either you can synthesize the siRNA molecule and incorporate in the liquid media and allow the fungus to grow in the lab, or you can go for the transformation method. You prepare a protoplast of your spore, conidia or whatever may be, and then the protoplast you go for PEG mediated. I have listed the method, no? PEG mediated method, polyethylene glycol mediated method. You make the construct using PEG. You transform the construct into the protoplast and then regenerate it. Then you will get the transformer. And the third method is you can make a silencing construct for the pathogen gene, but express in plant and allow the pathogen to colonize it. If it is a hemibiotrop or a biotroph, it will take the siRNA and then the target gene will be silenced.
Like one more question is like, do we have regulations for this plant expression of dsRNA?
See, you mean the guidelines, government regulations? Yeah, obviously. Even for the editing, which is not a non-GMO also, the people are little bit concerned. So that is always be there. Any transgenic,
No, at your lab it is done. Like plant
Yeah,
express
every
dsRNA
project.
you need to go for IBSC,
IBSC approval is
all the
needed.
approvals. Without approval, you cannot do this work, and you have to properly dispose the mutant also. You cannot spread to the environment.
There is a lecture on regulations of genome edited lines and genome modified lines by Dr. Ramcharan Bhattacharya. So you wait for that lecture. Okay, 1 hour lecture is there.
So one more like, is the study done on like systemic movement of dsRNA in fungal pathogens?
Is how it is translocation. No? So there is you can track it, but there is not much studies on this.
Thank you.
Yes, I think some good questions he asked. Anybody this side? Okay, this topic is very important topic because pathogenicity genes are something which is pertaining to pathogens. So we need to know what they are, how they are really doing the job. So I'm sure Dr. Shanmugam might have delivered a wonderful piece of information to all of you. Now that's only a tip of an iceberg. Right now you have to extrapolate it in different ways for extra readings. So reading is very important, right? Lectures alone, if you depend, definitely you will forget it by the time you leave this room. So you need to read continuously. Okay. So with that, thank you, Shanmugam,
Thank
for
you very much.
accepting our invitation.
Yeah.
And I'm sure this is a good lecture the overall context of this particular training. So I hope all of you enjoy it.
Yeah, take this as a motivation to work hard.
Definitely,
That's it.
So
Thank you.
I call upon Dr. Deepa to honor our speaker. So
Oh.
please come. So yeah, okay. So okay, thank you. So big clap. Okay, good. Now we will move on to the next agenda. Very important. Have you gone through the program? What is that? Okay, good. So under this project, it's not only the trainings, and we also deputed our students abroad just to get first-hand experience about the research what is going on in foreign laboratories and how they do research and the style of their functioning and their way of doing things. So we have two speakers today. They were granted this opportunity. They availed this opportunity. One who is going to make presentation first? Natasha Shayab. She was deputed to Australia, and she spent around 2.5 months in one of the leading laboratories under one of the very famous scientists. She will be revealing all those details now. The idea is what all her experience in the overseas lab that she wants to bring back to our country and try to implement in our system. So I want her to share her experience with all of you. That is the best way rather than me telling about she went there, she learned something. Let the words come from the horse mouth, right? Please come, yeah.
So, pointer, this one. Thank you. A very good afternoon, everyone. As Sir has mentioned, thank you, Sir, for the introduction. My name is Natasha Kashyap, and I'm a PhD third-year research scholar here in the Division of Plant Pathology, IARI. And I'm going to present my overseas deputation seminar for all of you. Probably you all might learn a few things or might be motivated to apply for PhDs or postdoc overseas after your degrees. So let's take you geographically a little bit from India to Australia. So I went for my training to the University of Southern Queensland in Brisbane, which is which is the capital of Queensland. So Australia, as you know, is a big country and a big continent, and it is divided into 6 major states and one Australian capital territory, that is Canberra. So the capital of Australia is Canberra. Most of the people think it's Sydney, but no, it's Canberra. So as you can see, Queensland, its capital is Brisbane, and just around a 2-hour ride from the Brisbane airport is this University of Southern Queensland in Toowoomba. It's a town in Queensland, Brisbane. So this is a new university. Although it established in 1967, it's emerging in its research perspectives, and the major centers or the major departments which are offering research in agricultural sector in this university are the Institute of Life Sciences and Environment, Center for Sustainable Agricultural Systems, Center for Agricultural Engineering, Center for Applied Climate Sciences, and Center for Crop Health. So I was the part of Center for Crop Health. It is a center in this university which is targeting mainly research in the improvement of crop health in terms of diseases, insects, and nematodes as well, pathogens and nematodes. So the Center for Crop Health is divided into different blocks: student facility, staff facility, which are separate, different laboratories like microbiology laboratories, different containment facilities, as we can see here as well, different glass houses, greenhouses, different types of incubation BODs, and a molecular biology block. So the interesting concept there in this university that I observed was every block was distinguished separately. It was not connected at all. So you have to, if you want to access any block, you have to go separately. You can't go from inside. The main, why this concept was important is so that you do not contaminate the other facilities as well. For example, if you're working in microbiology laboratory, after that you straightway have to go to your sanitation process. You have to completely change your lab coats, all your gears like your gloves, your shoes if you are exposed to the glass houses or the field, then you cannot directly enter into the containment facilities or the microbiology laboratories. So that indirectly you do not contaminate the laboratories or the fields or whatsoever. Next, talking a little bit about my overseas mentor. So I worked with Dr. Sambasivam Periyanan. So most of the students who are pathologists, they might be aware about the UG99 strain of rust, stem rust, which is a very, very important strain in rust. And he's famous for many things actually, but mainly in our field, specifically because he was the first person who identified and characterized SR33 gene analog, which is resistant to the stem rust of UG99. Additionally, he has many stars on his shoulder. However, he is editor of many reputed international journals, and you can look through his work for knowledge. He has worked on rust, wheat rust, stem rust, yellow rust, etc. And he's a plant pathologist as well as a plant breeder. Now, moving to the Center for Crop Health. So in Australia, the interesting concept for me was the funding agencies as well. So there, the main funding agencies in the agricultural research are two main governing funding agencies. One is the GRDC, which is Grain Research and Development Corporation, and another one is Broadcare Cropping Initiative, which is acronym for BACI. So this GRDC is an Australian government funded autonomous body which provides the funding as well as support for agricultural research in terms of institutional research as well as industrial research. And another one is this Broadcare Cropping Initiative, BACI. It also provides, it is a Queensland government-centered funding agency which gives funding to the at state level. So interesting concept from these is like they give funding, and every year they will organize research showcase in all the institutions which collaborate for industrial as well as institutional level research. So I was fortunate enough to experience this BACI research showcase in 2023, where the scientists, students and the staff as well, they presented their research, whatever they have done in that financial year, and how they made an impact in the industry and in the livelihood of the farmers and the people which are related to agricultural field. Interesting concept, they will always give you a sunscreen whenever you will attend any conference, because Australia is like the UV index of the country is very, very high. As most of you might know, the ozone layer has a hole which is directly above the Australia and New Zealand. So interesting. Now, going directly to what are the work that I carried out in the Center for Crop Health. So before you start in any foreign university, specifically in Australia, they are very, very particular about their biosafety guidelines. And therefore, before you're able to enter in any laboratory, you're allowed to enter in any laboratory, you have to undergo a series of set of protocols. Like first of all, you have to get an ID card. Without the ID card, you cannot access any building, any block. Let alone you can't even enter into the washrooms. So first you have to apply for the ID cards, and then you have to fill the SOP requests for the courses. So what kind of courses is that before even enrolling into the university or before even getting acquainted into the laboratories? So those courses are to get the acquainted and get a brief overview about the idea of the work and lab culture, and secondly about the chemicals that you are dealing with in the lab. Like what could be the hazardous nature of those chemicals and how you can handle those chemicals, and what are the chemicals that you should not mix at all at any cost. So these are the courses that you have to complete, following which you'll have lab safety induction training. So this lab safety induction training will be separate for each block of laboratory. For example, if you are a student who wants to work with microbes like pathogens and nematodes or any such things, you have to undergo a microbiology laboratory induction training. If you don't have any such work in the microbiology laboratories, you can just undergo the other facilities induction training, like containment facilities induction training. So for example, as I mentioned before also, if you are exposed to the field area, if you have work in field, you cannot enter any lab. You have to go to your room, take a shower, change your clothes, and then come back again and change all your work gear, and then only you are allowed to enter into another facility. So it's important to get induction training for all those separate laboratories. And one more very important thing that is in the universities and foreigners, they're very, very strict about the biosafety guidelines. So biosafety guidelines in terms of how they're managing the lab waste. They have separate dustbins for each type of waste that we are generating from the labs. For example, if it is like cancerous or if it is not, like for example, we are using gel and all those things, and if it is sharp objects. So they're very, very particular about disposing the waste in proper labeled dustbins. And if you fail to do so, there will be 2 times warning, and 3rd time they will take away your access cards and you will not be able to access into the laboratory or let alone in the university. So this is a brief about the first 15 days of my training, wherein I get acquainted with the university, I got acquainted with the funding agencies, attended a couple of research showcases, and I got inducted for getting access into the labs. Now before moving forward with my research work, I would like to give a brief idea about what kind of work or what is the related work that I am focusing on my PhD research. So I'm working on microRNAs, and the main part of my research is the microRNAs in regulation of host pathogen interactions. So I hope most of you might have heard about small interfering RNAs, as Dr. Shanmugam also told in his lecture just previously. So the counterpart of small interfering RNAs is microRNAs. Has anyone ever heard of microRNAs? With a show of hands, please. Okay. So microRNAs, as we know that in plants, in animals, or in any organism, there is a regulatory process which is referred to as post-transcriptional gene silencing. It is not only confined to plants, it is present in all organisms. So this post-transcriptional gene silencing can be through 2 main agents, as we can say. One is the small interfering RNA, as you can see on the right side of this picture, left side. Small interfering RNAs, as Dr. Shanmugam told, they can be exogenous. We can synthesize it in the laboratory as well, or they can be generated from the double-stranded RNAs, okay, where by the action of this dicer enzyme complex, which in turn leads to the mRNA cleavage, that is post-transcriptional gene silencing. MicroRNAs, on the other hand, what are these? MicroRNAs are endogenously synthesized small RNAs. Endogenously in the nucleus, they are synthesized from the pre-miRNA genes. These pre-miRNA genes inside the nucleus are acted by different enzymes, and these enzymes in turn transfer these pre-miRNA containing a preliminary stem loop structure. That's what's the characteristic feature of these microRNAs, and these microRNAs are transported into the cytoplasm. From there, it again is acted on dicer enzyme, and it acts for post-transcriptional gene silencing or the transcriptional repression of protein synthesis. So in both ways, siRNAs plus miRNAs act in post-transcriptional gene silencing. siRNAs are exogenously synthesized, however miRNAs, they are endogenously synthesized. So in terms of plant pathogen interaction, as we can see, there have been several reports where these microRNAs or siRNAs can travel cross-kingdom and lead to cross-kingdom RNA interference in the host pathogen interaction. So how? Like for example, from the pathogens, these siRNAs, because pathogens also, like many fungal pathogens, they produce these siRNAs, and these siRNAs are transported from the fungus or the organism into the plant cell, wherein this silence the host genes of the plants, which in turn can lead to susceptibility. Or on contrast, these microRNAs or siRNA can also be transferred from the plant to the pathogen, or they can directly be influenced by the action of the pathogens, which in turn can lead to susceptibility or resistance. So in cases where these microRNAs or siRNAs, they are transported from the pathogen to the plant, they can act as pathogen effectors, or they can act as plant defense factors or susceptibility factors if they are influenced by the pathogens. Many miRNAs, there has been reports with a typical characteristic length of 21 to 22 nucleotides. They have been found to be conserved for targeting the NLR genes. So NLR genes, as most of you might know, are the very important class of resistant genes in the plants. So these miRNAs, they act on the NLR genes and then lead to the post-transcriptional gene silencing of these NLR genes. And after the breakage of these NLR genes, the corresponding small siRNAs, as we can see, which are produced after the breakage of these NLR genes, act as siRNAs, means they again lead for the systemic silencing in the whole plant system. So the two question arises here. Number one is that can these pathogen miRNAs act as effectors for a very important pathogen pathogen interaction, wheat and Bipolaris solociniana, which is the spot blotch causing pathogen, which is my pathogen of interest. And the second one, can these pathogen induce the expression of regulatory plant microRNAs during the infection to suppress the defense related genes? With this, I already carried out a part of my research work here in India, wherein we already inoculated the seedlings of wheat with spore suspension of Bipolaris solociniana pathogen and collected samples at different time intervals, extracted the RNA and sent for sequencing, wherein we got the high throughput small RNA sequencing, Illumina based, and from there, after filtering and adapter sequencing, ligation and everything, all curation of the sequences, we got the final mature microRNA sequences. So after we have mature microRNA sequences, they can be identified as conserved microRNAs, conserved in the sense that they are present in a variety of plant species, or it could be novel microRNAs, novel in the sense that it is specific to a cultivar or specific to a particular plant species. So we identified many novel microRNAs as well as known microRNAs which are involved in this particular interaction. So after the identification of microRNAs and getting their sequences, the next step is to get the targets of these microRNAs. So for the identification of the targets of these microRNA, what we will do is, we will input the microRNA sequences into this. There are many softwares, like for example, I used Miranda software. There are many other software like PSRNA target software that you can use for getting the targets of the corresponding microRNAs. These are R software based software packages also, or some softwares are freely available on the web server as well. For example, a part of this Miranda software is available on a freely available web source, SRplot, where you can use it for the identification of the targets. After getting the fastest IDs of the targets and the sequences of the targets by the blasting of these targets, we will get the fastest sequences of these targets, which next step is to identify that what are the main pathways or the gene ontology of the genes which are targeted by these microRNAs. And for that, what we will do is we identify the metabolic pathways and the gene ontology of these target genes. There are again many R software based packages for this work, and some freely available web sources as well. For example, there is one Cas server. So for identification of the pathways of any unidentified or even identified gene, what you have to do is identify the KEGG pathway of that particular gene. KEGG pathway is nothing, it is a Kyoto Encyclopedia of Gene and Genome. It is a database which is created by the Kyoto University in Japan. So you just input your sequence of any particular gene of interest into that software, or maybe you can just add the KEGG number if you have a KEGG number, and from that you can get the pathway of that particular gene, what pathway it is involved in. For example, plant pathogen interaction, hormone signaling pathway, auxin signaling, etc. So you identify the pathways of these particular genes. This Cas server is freely available software web server that you can also use for the identification and getting the pathways of these genes. The next step is to get the gene ontology that you can again do on R software based packages, or you can do from the NCBI BLAST if you have less number of target genes. So with this, we identified the top 10 pathways of all the target genes which are involved in this miRNA target interaction. Next, you have to do the gene ontology enrichment analysis. So when you get the gene ontology of any particular set of genes, now you want to understand, because you have a large set of genes, you want to understand like how many, like what is the main gene ontology which is getting enriched in this particular set of genes, so that you target the only that is important in that interaction. So gene ontology enrichment analysis can be done with our software based packages as well. However, there are some free web servers online, like for example ShinyGeo. You just input your target genes ID or transcript ID or ensemble IDs into this software, and you can set the cutoff p-value, which is the just to avoid the error, which is kept minimum of 0.05% generally. So from there, we identified the key we did the gene ontology enrichment analysis, and we identified the main genes and what are their biological processes, what are the key cellular component that are enriched in that set of genes, and what are the molecular functions of those genes. Now, next step in this target identification is understanding the regulatory network of these microRNA and their target genes. Now, why is it important is because you get the microRNAs and then you have target genes. So how are they interacting, and how is this regulation taking place? You have to understand it, and then you can better do by the visualization of the network with the help of Cytoscape software. It is also a freely available software on the web server that you can download, and you have to feed in your microRNAs and your target genes, and you can customize it according to what are the things that you want to visualize in a particular network. So from this, we created a regulatory network for the microRNAs and the target genes, and we identified the main genes which are being main gene pathways and main gene functions which are being targeted by the microRNAs. Interestingly, which in our case was that most of the target genes were many protein, many MAPK site signaling genes, and many genes which are involved in signaling and resistance in the plant. Now, this was the work that I carried out. I was fortunate enough to have some other lab visits as well. As many of you might know, there is a Commonwealth Scientific and Industrial Research Organization in Australia, which is one of the leading research organizations in Australia, located in the capital, that is Canberra. And wherein I was fortunate enough to interact with Dr. Mingbo. He is a very prominent scientist working on RNA. And if you want to look at his publications, you can go through his ResearchGate and other related publications. He has done a lot of work on RNAs, and he was awarded with the Prime Minister Award of Science in 2012 or 13, I guess. So it was a very knowledgeable interaction and informative interaction with Dr. Mingbo. Next, I was also fortunate enough to interact with Dr. Anthony Miller. He's a scientist who is working on microRNAs from past 30 years of his career, and I had a detailed interaction with him wherein I cleared a lot of my doubts related to microRNAs, and he has a lot of publication related to the same field as well. And recently, interestingly, he gave me a research article that he did not publish at that time. It was a review article which is related to microRNAs in plant defense. And he was kind enough to give me that article before publication. It was submitted already, and only last month this is freely available on it is open access article. It is a very important article if you're interested in learning more about the microRNAs, and it is very descriptive review about the same. Now, my motto is work harder and enjoy more. So a little bit about the sites that I visited in Australia. So as I mentioned, Brisbane is the capital city of Queensland. So some of the sites that I visited in Brisbane, including the George King Tower and the Story Bridge, and very important Brisbane sign, and some things that I observed there. And University of Southern Queensland has the largest Japanese garden in Australia, which is very, very beautiful and definitely site for sore eyes. As we all know, Australia is a country of kangaroos, so I saw some kangaroos, baby kangaroos, koalas roaming in the university, and it was definitely some experience. Some other portions of the country that I was fortunate enough to visit was this island, which is second, North Stradbroke Island, second largest sand island in the world. And I visited the Parliament House, Canberra, War Memorial, and lastly, the most important, the Opera House and the Harbour Bridge, which is very, very famous in Sydney. And if you ever happen to go there, it's a must visit. I participated in some socio-cultural activities where Indians always find their communities, no matter where you go. And this is our whole Center for Crop Health team, where this was the last Christmas party. Where on Wednesday they will have a team morning breakfast session where they discuss about their work, personal lives, and just chit chat. So I was part of this as well, and that's the whole team for Center for Crop Health. I also tried some exotic cuisines, which was a bittersweet experience for me. But at the end of the day, Indians need our dal chawal, our sabzi roti. So. Lastly, how will I disseminate this knowledge and my skills? So as I did today, hopefully some of you might have learned from my experience and what I learned there. And secondly, definitely from the direct dissemination of my dissertation. And most importantly, what I learned was practicing the lab culture and biosafety practices in our lab. And all these bioinformatic skills that I have learned, I will definitely incorporate in other related work as well that I'm doing currently. And lastly, I would like to acknowledge the NAHP CAST PI, Dr. Vishwanathan sir, and for giving me this opportunity and providing me the fellowship for completing my training for 2.5 months. Then I would like to thank my overseas chairman, Dr. Sambasivam Periyanan, for his belief in me and allowing me to come to his country and university. Nextly, I am forever indebted to my supervisor, Dr. Malkhan Singh Gujjar, and my major member of advisory committee, Dr. Aunty Kumar sir, for this opportunity. And these are names of some of the people that were really, really helpful for me in terms of my research, in terms of getting acquainted in a new country. Thank you.
So that was our visit abroad through this project only, because we had different segments in this project. People went abroad, I think not only she, around 75 of them went from IRA, different part of the world. Any couple of questions? Not questions, maybe you expected something else, but what she delivered something else. If something is there, you can ask right now. Anybody? No?
Ma'am, I have one confusion regarding mRNA. You said mRNA is target map ke pathway. Like it's target for map ke pathway.
It can be.
I have one doubt. Like I have read one paper about alternate transcripts. Alternate transcripts also target map ke pathways.
Yes.
So how mRNAs are different from? How can we say like mRNAs are different from alternative transcripts?
Okay. So in plants,
Can we say mRNAs are alternative transcripts only?
yes. So I will tell you. So in plants, there is a regulatory system, okay, that wants to spare the plant's energy and does not want it to utilize at any place where it is not supposed to. So plants and every organism has some regulatory systems that prevent that extra wastage of energy, for example. So these microRNAs as well as alternate transcripts. MicroRNAs are preliminary sources for generating of these particular, as we can say, different type of transcripts, some chopped transcripts, which in turn can lead to systemic silencing in other host tissues as well. So microRNAs, the role is important in plant generally for regulation of its plant metabolic processes, which in turn is utilized or, as we can say, exploited in terms of plant pathogen interaction, abiotic stress, insect interaction, and a number of more.
That means during normal growth conditions, mRNAs are produced.
Yes.
However, during stress,
Yes.
they go for trade-off.
Yes. Actually, it depends. Constitutively, microRNAs will always be producing at some basal level of expression they will give. However, depending upon the condition, their expression will increase drastically, which in turn, yes, during stress of any kind, which in turn leads to the their particular target which is targeted, and then leading to the protein accumulation, difference in protein accumulation.
Thank you.
Okay.
Okay. Any more questions from anybody? No. Okay. If not, I think let us give a big hand to Natasha for sharing her experience. So thank you very much.
Thank you. Thank
Keep
you.
doing
everyone.
good work and disseminate what you learned, especially the biosafety things. We are very weak on that, but still we can improve all the time. Okay, good luck. Yeah, thank you so much. Yeah.
Thank you.
So that was the experience of Natasha. She visited Australia. Now let's listen from one more student who visited a completely different country, that is in Europe. Mr. Velmurugan, he visited Spain for the topic which is related to his PhD thesis directly. I think both of them did the same thing. Natasha was handling miRNA kind of work. She wanted to learn more. She went to one of the best laboratories in the same way. Velmurugan also went to one of the best labs who does the work which is matching with his PhD thesis project. Let us listen from him what he learned and whether something which we can also learn from him. Yeah, floor is yours. Yeah.
Hello everyone. Is it audible properly? So I'm Velmurugan, pursuing second year PhD plant pathology under the guidance of Dr. Andy Kumar. So I have visited to the country Spain for my overseas training program. The university I visited is the University of Malaga, which is a coastal city in the Spain. You can see that landmark. It's just above the African continent. This is the university's administrative building, which is at the heart of the city. This is a very old campus. It's a government university and established during 1972, and the enrolled students is more than 30,000 students. Okay, it's establishing the new campus that is just far away from the city, and it's in the outer ring of the city, and it has many schools. So the school I visited is the Institute of Mediterranean and Subtropical Horticulture. The institute mainly works on the crops like tomato, avocado, strawberry and olive. And in particular, I visited the lab that is Carrian's lab, headed by Dr. Victor J. Carrian. He is an assistant professor at the Department of Microbiology. You can see he's Dr. Victor Carrian. And they are the team members. So last year, in 2023 February, we had this team here. They were training us in a microbiome research under the NAGPCAS sponsor. So currently, he is having 4 postdoc students and 5 PhD, 3 interns and 1 technician. The lab mainly focus on the study of soil microbiome and plant microbe interactions, metagenomics and metabolomics, and also the study about the mechanisms of plant protection against the abiotic stresses mediated by the microorganisms, and also they study the diversity, dynamics and beneficial functions of microorganisms associated with the plants. I can say Victor is expert in his field and he is a rising star in the microbiome research. They have published many high papers in high-end journals. Recently, their team published a bioinformatics pipeline called BacLife in Nature Communication. With the genome sequence, it can tell the bacteria lifestyle, where it is living, whether it is pathogen or beneficial microbe like that. So coming to the purpose of my visit to the lab, I went to learn how to analyze the meta-barcoding data. So first we'll see what is barcoding. So barcoding is sequencing the genomic region for identification from a single species. Okay, so you will be isolating the microbes and you will isolate the DNA from a single microbe and you will amplify the particular gene and you will sequence it by Sanger sequencing technology, and by sequence analysis you will be getting that species identified. In the Sanger sequencing you will be getting the results as two reads like forward and reverse, and you will merge these two reads and you will be telling what organism it is. But in metagenome, so for example, take rhizosphere. In rhizosphere, it's a soil that is very closely attached to the roots of any plant, and it contains many organisms, bacteria, fungi, and many. So at a time, simultaneously, we will be identifying what are all the organisms present in that particular sample. So you can see in that barcoding, you can see the pointer, right? So in barcoding, during PCR amplification, only one gene from one species is amplified. But here, different genes, like not different genes, same genes from different organisms are amplified. And these amplified product is sent for sequencing. Here we are using next generation sequencing. Now the currently used technology for doing meta barcoding and metagenome is the Illumina sequencing. Okay, so after receiving the result and by doing sequence analysis, what are the multiple taxa or species present in that particular sample is identified. So to amplify a gene from different microorganisms at the same time, we need a universal primer. So that universal primer can amplify the same set of genes present in all organisms, and it is enough to identify and different organisms with its variability. So this is a 16S region of bacteria. We can see in this conserved regions are also there and variable regions are also there. These conserved regions are used to construct the primers. This conserved region is present in all the bacteria, same. And the variable region, there are 9 variable region, V1 to V9. V1 to V9. So these variable regions have the distinguishedness which will help in identification of the different organisms. For Illumina sequencing, we use this V3, V4 region, that is variable region 3 and variable region 4 of the bacteria, and it is sequenced using the Illumina platform, and this sequence is analyzed to confer what are all the species present in that particular sample. So the work I carried out in this institute is analyzing the 16S amplicon. So for that I used RStudio. In that I used a package called Dada 2. So Dada 2 is the latest version, and before that Dada was there. That is divisive amplicon denoising algorithm. Algorithm is a set of instructions given to perform a task. So what this algorithm will do, after receiving the sequences, we cannot say all the sequences we received is correct, 100% correct. There will be definitely some errors will be there. So to remove those error sequences, this algorithm is designed that will remove the error sequence. Why can't we do it manually like Sanger sequencing? In Sanger sequencing, we'll get only 2 reads per organism, and it is very easy for us to, it's not at all easy also, but it takes a lot of time to correct each and every base pair and to arrive at a correct sequence. Here, after the Illumina sequencing, we'll get millions of reads. So that is very, very cumbersome to analyze each and every sequence manually. So that's why we need a package, this kind of package like Dada 2. So what does it mean, package? This package is containing a set of scripts, we can say instruction, that will perform a different sequence correction, trimming, and many things it will do. And as a package, it completes the analysis from the start to the end. So the workflow is like this. the microbiome sampling. So you will, let us say, we'll collect sample from the rhizosphere, then we'll do DNA extraction, then we'll do the PCR amplification of the 16S region, the V3, V4, and then we'll send the amplicons for the sequencing. This is done by Illumina paired end sequencing. Paired end means both the reverse and the forward of the same sequences sequenced. So we'll get the sequence as a FASTQ files. You know FASTA files, right? The FASTA file will have our sequence and a name identifier of the sample. But in the FASTQ files, there is one more added component is there, that is the quality of the base. It is referred as Phred score. It's from the value from 0 to 40. So is given to each base. Okay. So that tells what is the trueness of the base call. If we'll consider if a base call is having the Phred score above than 30, it is really very good. Okay. So after we get the FASTQ files, we'll start the data to in RStudio. And I want to tell one more thing before. So you can see this is the RStudio, how it looks. Okay, it has 4 quadrants. Okay, this is the first quadrant, second quadrant, third quadrant, and fourth quadrant. In first quadrant is called as the source pane. In this, we'll just write the scripts, we'll give the instructions. And here, this is the console. Here, what it does, we'll come to know which program is running right now, whether that program is in progress or it is completed. And then this is the environment, whatever the objects we developed using the scripts like tables, Excel files, those things are displayed here. And this is the result portion. We can visualize the data in a graph or plots and etc. Okay, so we received the sequence data from the company, and we have to filter the forward and reverse sequence. We have to separate the reverse and forward sequences. Then we have to do the quality check. Quality check means whether the base call is the quality of the base calls we will check here. So this is how the quality plot looks like. You can see the green lines there. You can see the green lines, right? So that is the quality indication of the sequence. So from Illumina sequencing, we'll get a read length of 250 base pairs. The forward is 250 base pair and the reverse is 250 base pairs. So this is the quality of the read from 0 base to 250 base. And we can see, I already told above 30, the quality of the Phred score above 30 we consider as a good quality sequence. So when the green line drops below 30, these bases are not of good quality. So what we have to do, we have to remove those sequence. And this first box is of forward sequence and the second box is of reverse sequence. Always the forward sequence quality will be good and the reverse is not that good. So we have to just trim the less quality bases. So in this forward, after 240 bases, the last 10 base will be trimmed, and in reverse the last 50 or 60 is trimmed. Okay, so then moving further, and we will filter and trim the low quality bases. Then error estimation. What is error estimation? So this is the error estimation. In Illumina sequencing, the same sequence is read multiple times. That is called as depth. For example, if a same sequence you are sequencing it for that 100 times, at all the 100 times, for example, take the first base as A. Okay, so at all the 100 times, whether the first base A is read as A or other bases. Okay, so here we can see different boxes. This is how many times A is read as A, and here how many times A is read as G, how many times A is read as T, and how many times A is read as T. Like that, it is doing the error estimation. So it will take, for example, in 100 reads, if A is read as A for the 90 times, then in that place is really occupied by A only. So that is the error estimation. And you can see here, there is a red line and the black line. The line, this is the actual error, and the black points you can see now, this is the actual error, and this is the straight line in red color, it is the expected error. So in this case, our actual error and expected error are almost equal. So we can take this sequence forward. And after that error estimation, we will do sample inference. What is sample inference? The sample inference is, out of those millions of reads, it will tell what is the true sequences, unique true sequences. After that, we will merge both the forward and reverse reads. After merging it, we'll construct a ASV table, that is amplicon sequence variance. Before this ASV table, there is a concept it was used, that is OTU, that is called operational taxonomic unit. What is operational taxonomic unit? The operational taxonomic unit is, it is based on the sequence similarity. If the sequences, many sequences having more than 97% of similarity, means they are considered as a same group, okay? But that is not that distinguishing between two different species. That's why the later version is the amplicon sequence variant, which can distinguish two sequences with even one nucleotide difference, that is ASV table. After preparing the ASV table, we'll go to assign the taxonomy for each sequences, okay? So what is this sequence? This sequence represents which organism. So for that, we have to compare the sequences with the database like Silva, RDP and Green Genes. So everything is done by the package only, and then after that we can go up to species assignment also. So up to here the Data2 program, the Data2 package ends. It will take a lot of time if you are running with many samples. So few steps will take even 24 hours to complete the analysis. So consider the computer itself taking 24 hours, means how much time it will take for us to do everything manually. So if you want to analyze this kind of metagenomics data, it is better to have a good computer with a very high RAM capacity like 16 GB or 32 GB. That would be better. And after, with this species assignment, we can go for other analysis like phylogenetic tree construction. Then we can analyze the alpha diversity, then beta diversity, and also the differential abundance. So with this, the Data2 program is over. So with this Data2, we can do our analysis very rapidly and accurately, and we can get very high quality plots like this. It will be like eye-catching, and also the metagenome data analysis is now easy. So other than the lab visit, other lab visits like I visited Micropia. Micropia is a microbial museum which is located in the Netherlands. So where they display different kinds of microbes for the visitors. So I just kept few photos, but there is we just planned one day for Netherlands, and we almost spent 6 hours, 7 hours in this one museum only. It is a very small museum, but there is a lot to see there. So they have isolated microbiomes from various daily usage